Okay, good. So we're recording. Okay, so today we're going to talk a little bit about electrophysiology. Ask lots of questions. This is intended to be very open-ended. So it's not super structured. It's not like I have slides that actually depict everything that I intend to depict. Okay, so let me actually change this. So I think the first question when it comes to electrophysiology is what preparation are you going to use? What is it you're going to record? And the question there depends on what phenomenon are you trying to study. So say, for example, you want to record ion channels. Single ion channels are just something where you're really characterizing a conductance, something like that. So for that, there's systems like Xenopus oocytes, so frog eggs that you inject with RNA, and then you can poke them with electrodes and record, and then you can get relatively pure populations of high densities of ion channels. 293 cells, which are mammalian immortalized tumor cell line. There are also people who will actually purify protein and then put it in lipid and then actually paint it on this little thing with a hole in it. I've never actually done a person, but you can get very good recordings from that sort of thing. So that's kind of the lowest level, single ion channels sort of thing, or groups of ion channels. Then we can do neurons in vitro, cultured mammalian neurons, for example, or invertebrate neurons. And I think the question is there, well, what is it you're trying to study? And there are certain phenomena that you can study in cultured cells where it would be valid to study them, certain ones where it's not. So I feel I should definitely mention the squid axon. So those of us doing in vivo physiology in mammals, we need to stay a little humble and remember that the squid axon is still the source of the greatest discovery in the history of neuroscience. So make sure I'm actually recording here. Verify that. Because I'm actually not. Okay. Yeah. All right. So there's things like CFOST was discovered in cultured neurons, for example. So there's lots of things that could be done still in cultured neurons, but it's one of those things where you have to pick the right level for the question that you're studying. I think a lot of times people haven't. So you can do ex vivo neurons, which would be the most common thing for mammals would be slices. So you can either do acute slices where people will, perfuse an animal with ice cold saline, chop out its brain really quickly, and then put it in, bubbled, ACSF, artificial CSF. People can also culture slices for periods of time. Some people are doing turtle slices. It's actually fascinating because turtles apparently, or some species of turtle, have to survive without oxygen for long periods of time when they're underwater. So that if you take their brain slices and you put them in the refrigerator, you can come back a few days later and they're still totally fine. So I should say that with slices, so interesting things to know what slices. So hemoglobin carries a lot of oxygen, and there's no liquid that people have really come up with that really carries oxygen as well as hemoglobin. So hemoglobin is not just that it carries oxygen, but it carries oxygen to the site and then it releases it because of changes in pH and that sort of thing. So hemoglobin is very specifically designed for this, and people have never really been able to make a good artificial blood substitute. So there are some of these fluorine compounds people I think are starting to use, but hemoglobin carries lots of oxygen. So when people will bubble oxygen through ACSF and then put this oxygenated ACSF onto slices. So oxygenated ACSF has way less oxygen than actual brain in vivo does, far less. So it's not even close. So these neurons are operating in this regime where they're very starved of oxygen. And so historically, this is an interesting fact that I learned not that long ago. So when people first were doing slice recordings, they would actually extract CSF from rats. They would puncture, I think it was some sort of, posterior cerebellar cistern, and just collect a bunch of rat CSF, and they try to culture, and they try to cut acute slices and do slice recordings. And the slices would just go crazy and they would die. And the reason why is because there was too much activity. So when they made ACSF, the people who figured this out, ACSF is the ionic composition of ACSF is specifically designed to quiet down activity in a brain slice to make it so the slice takes less energy so that it can survive this hypoxic environment. So it's so I guess the thing with slices then is that there's certain questions I think you can answer in slices and certain ones where people probably have tried to answer certain questions in slices and maybe gotten false conclusions. So, but I mean, the sort of stuff you can measure in slices, of course, be, say, looking at things like detailed connectivity, which kind of cells map on, synapses on which other cells, characterization of synaptic physiology, things like that. But sometimes you see things where people are trying to model in vivo phenomena on slices, and it's hard to know exactly, things like hippocampal sharp wave ripples people model in slices, or epilepsy, like an epileptic slice. What exactly does that mean? And I think there's a little bit more gray area there. Then the next question, Victoria?
No question, no.
Okay. Can you mute your microphone, Victoria?
Yeah.
So I want to say I checked one of the videos. I think it was this last lecture just to make sure it was okay. And when anybody talks, then they become like their window becomes the whole screen. And so Victoria was cracking her knuckles right by the microphone. So the middle of my talk, what happened is it would just like pop to her face, and then all you see is her face, and you just hear her cracking her knuckles and just like looking really blasé. So I don't know.
I have a question.
Yes.
Does ex vivo pretty much in neuroscience always apply to slices, or are there other?
I mean, so that's the thing with words like in vitro and in vivo and stuff like that. It's sort of, some biochemists will say that in vivo means in a cell, and in vitro means, in something where water's not the solvent, like, proteins in benzene or something, right? So, I mean, I guess there's a sense in which cultured neurons are ex vivo, right? Because you're taking those neurons out of an animal's brain. So with the caveat that these terms are a little bit arbitrary and not everybody completely agrees, but at least you guys know what I mean. I mean, you take neurons out of the brain and then you record from them. Yeah, so slices, I mean, there are things like Drosophila, you can take the whole brain out. I'm sure that there are other preparations people have, like, I don't know, spinal cord or, invertebrate stuff. So I don't know, stomatogastric ganglion, I think probably would fall under this category from lobsters. But in any case, in the next level, so I should say one of the things about slices is that, so the ACSF is artificially designed to suppress activity. And then of course, the other thing is that you're removing all of the afferent inputs. So there's a lot of brain structures where they're relatively silent in slices because just they're not getting any inputs because everything's been cut. Another thing I should say about slices that just may be relevant is that they have a certain thickness. I think they're typically 300 microns or something. So I should say I'm not a slice physiologist. I've never done slices, so take everything I say with a grain of salt. But one of the things is that it can be difficult even to preserve connectivity between two sets of cells in the same slice. You have like two cells and then the axons go up and then come back down. If the plane of the slice can actually still cut the axons even for those cells. So I think people have worked on getting the angles right so you can get cells that are connected to each other, say cortex and thalamus, for example. Anyway, so the next step up is anesthetized animal. If you think about anesthetized animals, that's sort of a weird thing to do in neuroscience because anesthesia shuts off the brain. And then you're going to study the brain when you gave a medication that's sort of specifically designed to shut off the brain, and then you're going to study the brain. So obviously it's not that simple. I guess what I'll say is that there's different types of anesthesia. They have very different effects. And I think there's always caveats in studying stuff under anesthesia. I think I would say that the field historically has done too much work under anesthesia. And then now when people started doing in vivo work, they found the stuff actually looks very different. And that a lot of that anesthesia work, the results people drew from under anesthesia turned out not to generalize. So people don't do anesthetized animals much anymore, but there's still certain things I think it's probably good for. People now I think are moving more towards head-fixed animals. So what I should say, head-fixed monkeys have been in neuroscience forever. Head-fixed cats and monkeys and so forth in vision have been, the mainstay of vision research. And for vision in particular, you need to control what the animal's seeing, so you kind of need to fix the head. So that makes sense. So the head-fixed mouse prep is actually pretty recent. So rats do not tolerate head fixation super well. People have done it, but I think it's just hard to head fix rats. And so all the physiologists doing rats now, a lot of them are people moving to mice. So head fixed mice is much easier to do. And then head fixed mice has become more common. There are still certain things, I think there are still limitations to head fixed animals you still have to think about though. And I guess what I mean is that you have to think about the limitations of each of these systems and what phenomena you can't study in that system, what requires you to go the next level up. So an example for head-fixed animals is two-dimensional place cells in hippocampus, for example. That head-fixed animals do not have two-dimensional, if they're navigating a two-dimensional virtual environment, they don't have regular place cells. And the reason why is because like the head rotation and vestibular inputs play such an important role in generation of place cells, apparently, that you can't, that head-fixed animals don't have them. So that's just one example of something where you have to be careful what it is you're studying, make sure that your system has that thing. Then of course there's freely moving animals, which for a lot of things are really challenging. Obviously vision in freely moving animals would be extremely difficult. And just in general, a lot of things are harder when the animal's moving around. What I should have done, if I thought more about this, there's this awesome paper, this guy, Jose Delgado, right? This is a bit of an aside. So Jose Delgado wrote this amazing book called Physical Control of the Mind Toward a Psycho-Civilized Society. And this is book about, I think it's from the early 60s. And it's about all these crazy brain stimulation experiments he did with this notion that they were gonna solve all of society's woes and generate this utopian society. And it has all this crazy stuff in there where they have like, doing humans, deep brain stimulation, really weird stuff. And they have things where they have like a monkey with a deep brain stimulator and the other monkeys get to push the button to choose when to stimulate that monkey's brain. So then if they implant the alpha male, then the other monkeys get to push a button every time they disagree with his decision or something, and then it becomes a more democratic society. So anyway, there's lots of interesting stuff in there. The middle part, the sort of climax of the book, is he makes remote control brain stimulators and puts one in the striatum of a bull. And he goes to this, he's Spanish, and he goes to this big like bullfighting arena dressed up in a whole toreador outfit and has the bull charge him. He's waving the red cape, and then right at the last minute he pushes the button and the bull just like freezes like to a halt, turns to one side and doesn't hit him. Anyway, so that guy's a paper from 1970 in freely moving chimpanzees, 128 channel recordings, and freely moving chimpanzees with no wire. It's like wireless, with closed loop spindle detection and electrical stimulation. So in 1970, in a chimpanzee. So anyway, that was a very long-winded way of just mentioning something I thought was really cool. I'll put it on the GitHub thing for you to look at because it's great. But yeah, freely moving stuff. There's a lot more wireless stuff now. Anyway, so before I move on to types of electrodes, anybody have any questions about that? This is an important question, actually. What system do you use to study a particular question?
I have a question about, so for the ex vivo, you mentioned that the ACSF is designed to suppress activity. So I guess I'm thinking from the perspective of my project, where I expect like hyperexcitability. Are there other preparations that are used to study, or is it just like always kills the cells if you don't have that ability to suppress the slice?
So that's a good question. I mean, so I guess I'm talking about in animals like mice or humans or whatever, where the brain, if you have a stroke, for example, and your brain loses oxygen for a very brief period of time, then it gets damaged. And so there are other organisms like invertebrates or turtles or whatever that don't have that. But yeah, I guess what I'm saying is that slice recordings are always done under these massively hypoxic conditions. Even though people are like, I'm bubbling crazy oxygen through, it's not the same as if it had gotten blood. So it's energy, so you have to do something that suppresses the amount of energy that the tissue consumes.
Okay.
I think it may be somewhat debated, and some people deny this, or I don't know. And maybe I'm not 100% correct, but it is certainly the case that slices are not, that they're quieter than in vivo, and that ACSF decades ago was specifically designed for that purpose, and they're not getting enough oxygen or nutrients or what. I mean, so that's the other thing. They're getting, glucose from the bloodstream, oxygen from the bloodstream, all this stuff, and in a slice, they're not getting any of that.
What's like stopping them from using blood or artificial hemoglobin or something of that nature?
Well, so if you think about the way that hemoglobin and all that stuff works, is that it passes through blood vessels, right? It comes in through the artery and passes through the capillaries and goes up through the vein. So those capillaries are completely interwoven inside the tissue. So you know, there's no cell that's more than some certain distance away from a capillary. I don't know what that distance is. I don't know, 100 microns or something like that. But the distance that oxygen, you know, can diffuse through tissue and stuff is pretty short. So if you just have a slice, even if you just had a slice that was soaking in hemoglobin, it's not obvious that it would penetrate all the way to the inside. And then of course there's stuff like local changes in pH. So you know, so the blood pH drops as it goes from the arterial component through the capillaries into the veins. And that pH drop is part of what like causes the hemoglobin and oxygen to dissociate. So I mean, that blood and all that stuff's very complicated, and it's not something we can just. So I guess what I'm saying is, even if we, I don't know, we'd still have to somehow like hook up a blood vessel and a fake little heart and then be pumping stuff through it and all that. It's probably just not worth it. It's probably just better to figure out ways to do it in vivo, if that's the question that you're interested in, right? So like I'm saying, that lots of great work has been done in slices, and lots of important results can be gotten from slices, but you just have to be aware what the limitations are. All right, so now types of electrodes. So there's intracellular electrodes, which are, let me actually see. Okay, anyway, so we'll just go through them quickly and then I'll talk about them more in depth. So intracellular electrodes, meaning something that gives you access to the inside of a cell. So when you think about, you know, an action potential, where it goes up and then comes back down, that's the sort of thing that is, you know, intracellular recording. And the sort of oldest way that people have done it is with sharp electrodes. So and I'll talk about that, where it's just the glass pipette pulled to an extremely small point that you poke into the cell. There's patch electrodes, which are slightly different. I'll go over. And then now there's these sharpened tungsten electrodes that a few groups have made. It doesn't seem they've really caught on. And but I think it's one of those things where maybe in a few years, they'll refine this technology to the point where it becomes the next big thing. I don't really know. There's juxtacellular / loose patch. I'll explain that too, but that's something where you take an electrode and you put it up. It's like a glass type electrode, and you put it right up to one cell. So you're still outside the cell, but you've essentially still isolated just one cell. And you can fill cells with dye that way. So it's maybe just like a little easier than intracellular, slightly different than extracellular. So I should say maybe extracellular unit recordings, meaning you can record action potentials. So there's lots of different types of metal wires, which people have used historically. You just put wires into the brain. And then there's multi-site electrodes, which a stereotrode would be if you take two wires and then you twist them, you cut off the ends, so then you have two wires that are right next to each other. A tetra would be four wires right next to each other. And the idea behind those is that, so if you just put in one wire, the idea is that that wire will record action potentials around it. Whereas when you put in multiple wires, say four, like a tetra, 4 wires in a bundle, that each wire records the same cell. So you see this, well, I'll go over that in a minute. And then you can do with both spikes. And then there's field potentials, which are the local field potentials, which is when you take just sort of like a large wire, large electrode and put it in, or you record EEG, for example, from a big electrode stuck on the surface of the brain, you know, intracortical, usually on the scalp, which is averaging over a pretty large area. Right, so intracellular electrodes are generally made from, you have a glass tube, and you have a thing like, this is just an example, there's different kinds of pullers, but the idea here is that these things clamp on the glass and pull outward, and then you have a metal filament inside that heats up and then until the glass melts, and then it pulls it apart and then snaps it. And so this is the kind of thing where there's these pullers that get super complicated, and they have all sorts of parameters of like how you pull, and you know how much air to puff on, and how you heat the filament and all this sort of stuff. So you can make it so that you can customize and make perfect tip geometry for what you're trying to do. So that's an example of what one of them looks like. You can see that it's, you know, it comes down to a very small point. Anyway. So types of intracellular recordings. I don't know if I'm just going to draw this. I'm going to see how this works. Okay. So sharp electrode, like I mentioned, is something where you have electrode like that comes to a very small point, and then you poke it inside of a cell. And then you have a wire here inside there, and typically it's a wire it's usually silver that's been chlorided, so you can soak it in bleach, or you can do it electrically so that you have the silver chloride coating on the outside, and you have your solution inside there. And so typically if you're doing an intracellular recording, you would want to have the solution inside your pipette to be similar ionic composition to the intracellular, cytoplasm. And so you poke inside the cell and you do a recording, and then you have some sort of ground wire outside. And we'll talk about grounding and referencing later on. Okay, so this would be sharp electrode. Okay, then you have patch clamp electrode. So for patching, it's something where it comes to a wider tip. And so the sharp electrode, when the electrodes break, they break and they have a sharp glass end. And sometimes people will sharpen it even more to angles and whatever. With patch clamp electrodes, what people do is you have a slightly larger tip diameter, and then people typically fire polish it, where you have under the microscope, you go close to a heating element, you just turn the heating element on so that the very tip gets very smooth. And then you bring it down onto the surface of a cell, and then apply suction, and then it sucks a piece of the membrane up into it like that, and it forms right there what's called a gigaseal. Which means like a giga-ohm resistance seal. And so this configuration where the cell is still intact and you're just taking like a little, you've patched onto a patch of membrane there, you formed your gigaseal. So this would be called cell attached. Okay, and then if you apply suction to rupture that patch, then that's called whole cell. Okay, so cell attached, I guess, is extracellular recording, and whole cell is intracellular. There's also something called perforated patch. Well, all right, so let me back up for a second. So the thing with sharp electrode recordings, to some extent, you're changing the ionic composition of what's inside here, but not fully. At least that's my understanding, and I don't do these types of recordings, but you can load dye into the cells and stuff like that. But with whole cell recordings, where you rupture that patch near this large opening, then you're completely replacing essentially the contents of the cell with the contents of your recording pipette. So that you might have to put like GTP in there if you're studying some GTP dependent protein or these sorts of things, and so you're changing it. So it's possible that the properties of what you're studying might change because you've changed the contents of the cytoplasm. So if you're concerned about that, there's a technique that is called perforated patch. The idea there is that people have a patch electrode where they have the wire up there, and then you have your solution, your main solution up there, and then the very, very tip they fill with a solution that's full of this. Usually they use these things called gramocidins or some other. They're like tiny little ion channels that they form ion channels in the presence of lipids, and you patch onto the cell, and then they go and they actually make ion channels there, and then you are doing a cell attached type geometry, but you've actually gotten access to the intracellular compartment through these things that only allow ions to diffuse through. People don't do that very often. That's apparently very difficult to do. Okay, so that's cell attached versus whole cell. So differences here. So patch clamp like this is more mechanically stable because you've really made a seal on there. And sometimes even if it drifts a little bit, you can see that it pulls the cell off with it. And that's more mechanically stable than sharp electrodes. So sharp electrodes, you can poke multiple cells in one animal. You can poke, record a cell, keep driving it through and poke more cells. Patch clamp electrodes, once you've made a seal, then you have to replace the electrode because it's essentially fouled the end of the glass. So you can do blind sharp recordings pretty easily, I think, but blind patching is very difficult. So typically that's done in slices, for example, where people are looking at where they can see it with a microscope and then they directly patch onto a cell. Whereas sharp electrode recordings are easier to do in vivo, and you can just have a micro drive that moves it. So there's one issue with patch clamp recordings, especially in vivo, that I think a lot of people are not aware of, that is a bigger issue than people maybe should be more aware of it than they are. And that's that if you have so in here you have a high potassium concentration, right? So that means that in your solution out here, you also have to have a high potassium concentration because that's what's going to mimic the cytoplasm once you patch on and then rupture the patch. So what happens is that, so if this is the brain and then the cell you want to patch is down there, then what happens is you have your electrode coming in like this, and you have to apply outward pressure the entire time you're going down to keep the tip from clogging until you get down to the cell, and then go onto the surface, and then see your resistance start to go up, and then you change it to suction to form the seal. So what that means is that the entire time you're going down, there's this high potassium solution that's coming out of your tip. And so high potassium extracellular solutions increase activity. And so what happens is that all these cells start firing like crazy all around the site as you're going down, and then finally you patch onto a cell. So people typically in vivo will patch onto a cell, and they have all this like almost epileptic activity that has to die down, and they have to wait for 30 minutes or something before they record. And so it's, you know, I don't know how big of a difference it makes, but it's something I think a lot of people aren't aware of. So sharp electrode recordings, you could argue that they cause less perturbation of the system. Okay, so then why do people do whole cell recordings versus sharp electrodes? What's the advantage? So the big advantage to whole cell recordings is the access resistance. So actually, let me see. Okay, so now we've got whole cell. Okay, so I've got a whole cell recording there. So by access resistance, I mean that this is your electrode here in the solution. And if you imagine that from there, you still have it's essentially like a resistor, the resistance between, electrical resistance between your electrode here and then the inside of the cell. It's called access resistance. So in sharp electrode recordings, the access resistance is very high. And then you also don't have as good of a seal there. Whereas in these recordings, you have a tight seal there. And then so the resistance is, you know, in the gigohm range around it, and then your access resistance is much lower. So what that means is that you can pass current through this electrode. So if you pass current through a sharp electrode, which you can pass current through the electrode, but then because there'll be a drop across that resistance there, then you can't record voltages and pass current at the same time. So you can either do one or the other. And so for actually large cells, say like xenopus or some cells in invertebrates, people will do what's called 2-electrode voltage clamp, where they'll have 2 sharp electrodes on the same cell, and they'll put one of them close to the center, one of them close to the membrane. And so this one here records the voltage, and then this one injects current. And that way you can do voltage clamp, but you have to have 2 electrodes per cell, because you can't. So there is this thing called, you can do discontinuous single electrode voltage clamp with it, but nobody does that because it doesn't really work. And so you basically have to have 2 electrode voltage clamp if you're going to do voltage clamp through a sharp electrode. With patch electrode, you can do voltage clamp with just one, you know, with just one electrode. So people can patch on, and they can drop the, you know, membrane potential down to some certain, you know, potential, and then record synaptic inputs at that potential, and then change it to a different potential and record inputs there. So voltage clamp versus current clamp, I guess I should talk about. So voltage clamp, so current clamp isn't really clamping anything. It's just that you are passing some amount of current. And if you think about a passive recording, that's a current clamp recording where you're passing zero current, or you can kind of, you know, just inject certain amounts of current. Whereas with a voltage clamp, it's a feedback regulated thing where you essentially are where you are as a feedback circuit that measures the voltage and then injects the right amount of current to make the voltage stay where you want it to be. And then you measure how much current it takes to keep the voltage at that level. And so that's a way of measuring synaptic currents. I believe it was Hodgkin-Huxley who developed that for squid axons. I think they were the first ones. All right, so I think that's mostly the stuff about intracellular recordings. I guess other advantages, and this stuff I don't know as well, but I think is 90% true, that there's certain, for example, channel blockers that if you do with a whole cell electrode, then you're replacing the contents of the cell better, so you can put in some sort of ion channel blocker that blocks the inside of channels. And then whereas it's hard to do that with certain blockers and stuff with sharp electrodes, I guess, because not enough gets in. Anyway, so that's the 2 main types of intracellular electrodes that people use. Questions before I move on? right? Talked about these, composition of pipette solutions, replacing the cytoplasm, mechanical stability. Okay, so, oh, and then this. So this is something that you can electrolytically sharpen tungsten. So if you have, and I've done this before, it's like, I think it's like if you have sodium hydroxide or something, and then you put an electrode in it and have an electrode on your tungsten, and then you repeatedly put the tungsten up and down, then it essentially will electrolytically remove the tungsten and sharpen it to this incredibly sharp point. So you can do that to make like tiny scalpels so you can slice open a Drosophila cuticle, things like that. But people have done stuff with this with insulators, and they can somehow make these metal tipped electrodes you can poke, and it will actually, you know, somehow that part enters the cell, and then that forms a seal in some way so that you can get access to the intracellular compartment. And so for whatever reason, this hasn't really taken off. So I think it's not ready for prime time yet, but that's something you might see in the future. And if this works, so I should say one of the problems with patching cells is that it's very hard to do, in the sense that you have to, it's like this mystical skill, you know, that these people develop, that you have to have the thing in your mouth and approach and then push and then suck and then do all this stuff. They have robots that can do it now, but still you can't do it from lots of cells. Whereas if you had something like this, you could make an array of 100 of these things and put it into the brain and get intracellular access to a whole bunch of cells simultaneously. So if this works, it could, you know, be a big advantage. All right, so extracellular recording. So the first thing to recognize is that the waveform is going to be upside down. So everything you see is upside down from what you're used to, because you're, so here's an example of something where they did simultaneous intracellular and extracellular from the same cell. And then they poke that cell and they have a tetrode outside, and you can see that's right there. That's the, you know, that's the action potential as you're used to seeing. And then that's what the extracellular trace looks like. And the scale of these is also different. So I think what this is that, so that's the scale for the, that's the scale for that one, and that's scale for that one. As you can see, it's 30 microvolts, that bar. So it's microvolts, not millivolts. So it's a lot smaller. Yeah, almost minus derivative intracellular, I guess. So the point is that, you know, from extracellular, you don't really do, so intracellular recordings, people do these sorts of things about, you know, analyzing the waveforms so that you can look at different currents and whatever. You don't really do that with extracellular. The only thing you can really do that is somewhat useful is that certain classes of interneurons can be distinguished because they have narrower action potential. So be PV positive fast spiking cells in the telencephalon, and about 40% of interneurons are PV positive. So actually, this is one thing you'll see all these papers where they say the interneurons do this, and then the pyramidal cells, whatever, do this. They're really referring to that 40% of interneurons that have the narrow action potential waveform, and then the rest of them are indistinguishable from pyramidal cells based on waveform characteristics. So, all right, so microwire recordings. What do I have next? Okay, so this is sort of the oldest way people have done it, and that's just you have a wire, right? And you have some sort of wire that has insulation on it, and then you cut it so that you have this very thin layer of insulation, and just the tip is exposed, and then you stick that into the brain. And there's different kinds of wires that people use that have different properties. I don't think people really use stainless steel too much anymore. The main ones that people still use, tungsten is probably the biggest one. And then there's also nichrome. Oops. There's also nichrome. There's a nickel chrome alloy. So the biggest difference between tungsten and nichrome, and then stainless steel and these different ones, is the stiffness of the metal. So I'll talk about that a little later, but there's a trade-off between how, so if you have very stiff metal, it just goes straight into the brain and it pokes right exactly where you want it to go. Actually, I guess I'll talk about it now. And so a stiff electrode, you can target it very precisely to the location you want. But the problem is the brain is very not stiff, right? So what happens, you poke an electrode and the brain will sort of move relative to the stiff electrode. And so mechanical stability is less with the stiffer electrode. Whereas if you have a less stiff electrode, so nichrome is less stiff than tungsten for the same diameter, those wires, you have to sort of insert it in more. If you just stick it in straight, it'll just sort of go up. You have to sort of insert it more slowly, and you can still kind of, but it might still deviate more. But then once it's in, then then because it's more flexible, that it moves with the brain a little bit more when the brain moves inside the skull, and then you have more mechanical stability. So that's sort of for all extracellular recordings. So silicon probes are the stiffest of all. So they just plow straight through the brain like a knife, and then they have least stability when you're, especially if you're doing acute recordings. You'll have lots of what's called drift. Drift meaning mechanical drift of the brain relative to the electrode site. So with silicon probes, there's ways you can actually correct for drift, which is kind of interesting. But, right, so tungsten versus nichrome. So tungsten is basically inert. It's biologically inert. I mean that it's biocompatible, whereas nichrome's toxic. So what you have to do is you have to gold plate. You gold plate the tip. So gold is very biocompatible. So you can gold plate tungsten if you want to, or you can. And a lot of people actually do because it'll decrease your impedance. And so I think I have a separate one for tip impedance. We'll talk about it here. So impedance, similar to resistance, but at a frequency dependent resistance. So if you have a small diameter wire, then it's going to be higher impedance, and then a larger diameter wire is going to be lower impedance. So the impedance largely just controls sort of how far away from the tip can you see stuff. Like, how sensitive is your microphone? So if you if you're doing just recordings with a single wire like that, people typically historically will use fairly high impedance electrode because they want to record from one neuron at a time. So if you have a very low impedance electrode, you're just going to see all these spikes and you don't know which neurons are coming from. Whereas with a high impedance electrode, you can just get it up close to just one cell and just get really just one cell. However, you can also, with the low impedance electrode, you can sometimes see, say that you have two cells and their active potentials look different, so you can sort them based on that, for example. So there's somewhat of a trade-off there. So one thing I'll say about this, well, maybe I'll talk about that later. I'll talk about it now. So the big problem with this type of approach, so this is what people did in like primates in the 80s and so forth, and 70s and 90s, is that you're essentially isolating your single units based on the high impedance of your electrode tip. So what they would do, for example, in the visual system, was they would stick an electrode in and then kind of wave a flashlight in monkey's eye, and they listen to it, and they're like, I don't hear anything. They will further and like, oh yeah, I got a good cell. And then they leave the electrode there, and then they record, they show the stimuli and stuff. And so they fish around for cells. And so what happened with that is that people didn't realize it until years later, but there was this massive bias for high firing rate neurons that was induced by that. And so there's a lot of these, old papers where it's like, oh, the average firing rate of a cell in cortex is 30 hertz and blah, blah, blah. And it's like, that is not true. It's like 0.1 hertz basically. So there's all these cells that don't fire, most of the cells in the brain don't fire that often. And it's a distribution with a heavy tail where you kind of have a small group of neurons that fire a lot. And so people have historically oversampled the high firing rate cells and gotten some conclusions that probably weren't as reliable as what they thought. This has one drawback to this approach. All right, so tip impedance, we talked about that. If your tip impedance gets low enough, so if your wire is big enough, then you're actually just recording local field potentials. That you kind of don't see spikes at all, and you're just recording like a big average voltage across a larger area. Okay, so tetrodes. So tetrodes were the big breakthrough, and I think it was in the 90s. They were actually first developed for monkeys, and then they didn't take off as much in the monkey field as they did in rats. So rat people kind of largely all switched to tetrodes. And so part of the reason why is that people studying rat CA1, so how hard it is to isolate one cell from another depends on how densely packed the neurons are. So there's places like CA1 pyramidal layer in hippocampus where the neurons are packed incredibly densely into this tiny little layer. And so spike isolation, I think or single unit isolation was more difficult there than it was, for example, in primate V1 where the cells are spaced farther apart. So I think that's part of the reason that this technology was more widely embraced by, hippocampal people as opposed to say V1 people or some other areas. But the idea here is that you have a tetrode, which is 4 wires twisted in a bundle. So what you do is you have this very thin wire, and then you have a little thing that spins it, and then you have a hot air gun, and then you blow on it, and then the insulation melts just a tiny bit, and then they all stick together, and then you clip it, and you have your tetrode. And so here, the idea is that you're recording from a bunch of different cells, but the idea is that each, so if you have 4 sites, so you might look like this, where one's kind of like that, another one's like that, and then this one's like that, and then that one's like that. And so that would be cell A, right? And then cell B might be like this. And so you essentially can record more than one cell from the same tetrode, but because you're sampling each cell from 4 different, slightly different locations in space, then they'll have different profiles across those 4 sites, and then you can say, oh, this is cell A and that's cell B. And that process is called spike sorting. So I should say that spike sorting is never 100% reliable. There's no such thing as 100% reliable spike sorting. So if you have some sort of experiment that requires you to really isolate individual neurons, and that is super important, you get just one neuron, you have to do intracellular, or you can do juxtacellular, or you could do maybe the thing with, like I mentioned, with a super high impedance electrode. And I realized I just forgot to talk about juxtacellular recording before going to this. Juxtacellular is a similar sort of thing where you just have a glass electrode and you get it really close up to one cell. And then you're isolating just one cell. So it's still extracellular, but it's with an intracellular type electrode that's very close to the cell. Right. So the idea here, though, is that you have more sites, and multiple sites allows you to essentially triangulate and figure out which cell you're recording from. Right. So remember before I told you that is beneficial to have a high impedance because that allows you to isolate individual neurons. Right. So that's if you have 1 wire, that's true. But now if you have 4 wires, it's always better to have a lower impedance. And that's some people debate that, but those people are wrong, right? It's always better to have a low impedance, because what happens then, you just record from a larger and larger radius around the electrode, and you might get more and more unsortable stuff, but still the amount of well-sorted units you have will always go up if you have lower and lower impedance. So you want to get the impedance as low as you can. So with tetrodes, what you do is you gold plate the tetrode tips, but what happens if you gold plate it too much, then they'll actually plate over and then actually connect to each other and then form shorts between the tetrodes. You want to plate up to that point, but not beyond that. And there are all sorts of crazy tricks people have for gold plating that can increase the surface area by adding these dopants to the plating solution, whatever. But that's the idea, is you want to gold plate them. So like I said, if they're nichrome tetrodes, you gold plate them partially for biocompatibility, but anyway, you still want to decrease the impedance, so you probably gold plate any tetrode. Right?
Luke, I have a question.
Yeah.
So with the low impedance electrode, does it mean it's also more susceptible to noise, or it doesn't matter?
So it's actually, believe it or not, it's actually high impedance electrodes are actually more sensitive to noise from external sources. I guess it depends what you mean by noise. So the high impedance electrodes are more sensitive to artifactual electrical noise than low impedance ones are. And so I'll talk about that a little bit when it's the silicon probes. The silicon probes have much higher impedance, and so tricks had to be figured out in order to decrease electrical noise, it turns out, when you record a high impedance electrode. Okay, so, all right, so this is an example. Sorry, someone else have a question? Okay, anyway, this is just an example. This is a model that someone made that's found on that website, but the idea here is that you have is that at different locations in space, the cell will look different. The action potential looks different. So that way, essentially, if you sample multiple points in space, then it's exactly what I just explained to you before. And so here, if that's what the different cells' action potentials look like, that's an example of what real data would look like, and you can sort them. And so getting 6 cells from one tetrode would be pretty good, but I mean, people get that many cells from one tetrode. So you can get more cells than you have sites. And you do that from a combination of looking at the distribution, essentially, of amplitudes across the sites, and then also looking at the shape of each individual action potential. And we'll talk more about that probably next week for spike sorting. I'm not going to be here next week, by the way, but people from my lab will give that lecture. All right, so next came silicon probes. And probes and tetrodes have their pros and cons. But so this is an example of one shank from an 8-shank probe. So I guess they have 8 sites per shank. So probe, actually maybe I'll show you. So that's an example of what probes look like. Like they have lots of different geometries you can choose. And so this would be an example of this is probably similar to the one that they were using. So you have an 8 shank probe, and then you have 8 sites per shank. So instead of being a tetrode, you could think of each one as maybe being more like an octrode or something. And then you spike sort each one individually. And so if you look at, so that's an example of data where they have 64 channels. You can see there's some local field potential, fluctuations, but you can see all these different units. And you can see pretty clearly, for example, that, so I should say that it's almost never the case that the same cell will be detectable on more than one shank. So the shank spacing here is typically like 200 microns. So so you just look at each one individually. So you can see, for example, how that one is different than say this one, right? So those would be 2 separate cells recorded on the same shank, or that one's different from that one. So you don't do this manually. There's programs that do this, and you have to go back and then curate the automated output. So people do manually spike sort, I should say. Some people still do for tetrodes, but once you start getting more sites per shank, then it becomes sort of undoable to do manual sorting, and you have to do automated sorting. Right, so advantages of this, I should say, over tetrodes. So one advantage is that it's just easier, because you don't have, making tetrodes takes a long time. So you have to make tetrodes, and you have to, gold plate them and cut them, and you have to assemble the whole thing, and it takes a long time. Whereas silicon probes are much faster. You take the probe out, you glue it onto a thing, and then you're ready to go. Probes are a lot more expensive, so that's the other thing. So silicon probes can cost $1,000, maybe up to $3,000 for the expensive ones. They're very fragile and easy to break. So there's a trade-off there. Of course, if you take a week of a person's time, that's probably worth, say, $1,000. So then it's a whole trade-off. And there's pros and cons to each. So the one thing that's about silicon probes is that the precise spatial distribution of sites is known, unlike a tetrode. So a tetrode, you just have 4 sites, and you don't really know where they are relative to each other. Whereas here, so that is somewhat useful, that this information of where those sites are. So an example would be sort of that if you are recording, say, in CA1 parameter layer, and you have, say, the parameter layer is there, then you can say, look down here, and you'll see the sharp wave from a sharp wave ripple, and then up there, you'll see the ripple oscillation. So these were primarily designed for use in hippocampus, because you have this laminar architecture where it's actually important to know exactly, where things are.
Sorry,
That's why these.
Oh, sorry.
Yeah.
So can you not reuse these silicon probes in any case?
You can reuse them until you break them.
Okay.
And so how good you are at not breaking them is sort of, so then that becomes the skill.
Breaking them physically, or like, can you tell by the
physically break them? Yes.
Okay.
Yeah, they're super fragile, and really fragile and really expensive. So when people first started working with them, it's just like they're just completely panicking, and they have this $3,000 thing, and then you just touch it, and then it snaps. So yeah, so you just have to get used to the idea that eventually everybody breaks some, and you just break as few as you can, and you be careful. But yeah, they break.
Thank you.
You have to clean them and stuff.
Luke, the slide at the back, what's the yellow probe with the, if you go back to your other slide where you talked about electrodes?
I'm sorry, no, which slide you mean? Which slide?
The examples of the electrodes.
This one?
Yeah, what's the yellow one? Is it a shank electrode or a different?
So that to me looks, so there's also people can make flexible electrodes that you can put on the brain surface. And then those probably aren't, that's probably not silicone, that's probably this stuff called parylene, which is like this flexible plastic. So that's my guess as to what that is. People do that a lot for brain surface recordings.
Okay.
And you can get isolated units from that down to like layer 2, 3, but not below that. Layer 2, 3 in a mouse or a rat, but not lower than that. That one maybe you can't, but people have made ones where you can. They're not commercially available, I don't think. It's like there's engineering labs that do that, and then they're the only ones who can make these things. Because making these things, it's like clean room, PhD in electrical engineering level stuff. It's still the sort of thing that compared to the chips in your iPhone, this is technology from the 1970s or something, as far as actual silicon microchip stuff, but it's still not something you can do yourself. So so NeuroPixels is kind of the newest ones, you can say. most of you, or some of you probably heard of NeuroPixel. So their geometry, well, let me back up for a second. So the pros and cons of probes versus tetrodes is the probes have this very precise geometry. And so what I was saying, for example, one like this is probably made for something like hippocampus where you have CA1 going through like that, and you want to be able to record from there and there. Or if you have, say, cortex and you want to record, you know, so say you want to stick it in cortex like that and you want to be able to see the different layers and say, oh, cells in layer 4 are doing this and cells in layer 1 are doing this. That's the advantage of that. Whereas with tetrodes, you just kind of stick a bunch of wires in and you don't really know where they are. However, there's certain things like if you wanted to record from, with tetrodes, typically your unit yield is higher if each tetrode is independently movable, because if one tetrode's in a bad location, you move it to a good location. And so you can optimize it. The total number of cells you'll get with tetrodes typically is higher for the same number of sites. So if you had 16 tetrodes for 60, which would be 64 recording sites, then you'd probably get a higher unit yield than a 64-site silicon probe. So it depends on your question. Another thing that tetrodes are very valuable for is if you want to record from multiple regions simultaneously. So what people can do is they can sort of design their drive such that, you know, so imagine that's the mouse skull, for example. And then you sort of have your tetrode thing where you have a bunch of ones up there in like, you know, PFC, and then you have some ones back there in accumbens, or I'm sorry, hippocampus, something like that. And then your tetrodes can poke down, and then you can record from both those things at the same time. And that's much harder to do with a probe because you can't make a probe so you'd have to put 2 probes on, and then it gets too big for the animal's head and that sort of stuff. So tetrodes are more flexible in terms of what you can do with them with multi-region recordings, and then each one being independently movable. But then also you don't know, and when they go in and they're packed closely together like that, then they all just kind of go off in slightly different directions, and you actually don't, it's, I think, almost impossible for people to distinguish which one of those went where. Whereas with probes, you know exactly, that site is exactly however many, you know, millimeters away from that site. So pros and cons of both.
Luke?
Yeah.
You speak a lot about how electrodes sometimes like don't necessarily go perfectly vertically, but go into places that you can't really control.
Mm-hmm.
Can you speak about the reliable methods that are available to us to check where we were recording? I'm thinking coding with DI, but there are probably other stuff.
Yeah, so so yeah, the main way, that's a good question. So how do you know where your electrodes went? I wish I had some slides on that. So so the basic idea, let's see if we can. Put it on here. This, I'm going to save this. So that's the reason why you can save the ink drawings. I learned recently. Anyway, okay. So the idea there is that, so say you have, I don't know, the brain from the side. I'm very mouse centric, right? And then you have your electrode that pokes down in there. So you can code it with something like dye, which is a lipophilic dye that you paint on, and then it dries, and then it deposits, and you'll have this sort of dye channel around there. The II diffuses away in a couple of weeks. There's one called CM dye, which has some sort of extra stuff attached to it that makes it so that it doesn't diffuse away from the site. So for chronic recordings, you can use that. There's retro beads. You can paint on these fluorescent latex spheres onto, let it dry, and then poke that in. The problem with that approach, there's 2 problems. I mean, so that approach works. The problems are, number one, sometimes it's hard to know. You can see the channel, but you can't see exactly where it ends. So you know your electrode is there, but you can't tell, was the tip here, was it there, was it there? And so what you typically do then is you try to place an electrolytic lesion. So you stick an electrode somewhere, usually in the animal's rectum, and then you pass current through this one until you make a little burn there. And then often the combination of those 2 things, where you have your dye channel and then your little burn site at the end, then you can sort of know where your electrode went. So the problem with that, however, with tetrodes, is that if you do 1 tetrode, that's not a problem, because you can see exactly where your 1 tetrode went. The problem is if you have 16 tetrodes packed into a tiny spot, you know, right next to each other, then you don't know which one's which, right? So that's what I'm saying. If you have 16 dye channels and 16 burns, then you just don't know. So it makes it harder to localize anatomically. with probes, silicon probes, the nice thing, of course, is that if you know where shank number 1 is and you know where shank number 4 is, then you know exactly where shanks 2 and 3 are because the geometry is completely fixed and predictable. So yeah, you can use different colors of dye. People now, we're actually building one for the department. So how you actually image your dye channel in the brain. The way it's typically done historically is, you know, you fix the brain, you cut it out, and you section it, and then you image the sections, and you try to make some sort of crude estimation. So people start having software where you can kind of draw, where you can label markers, and then it can put it rendered back into a 3D space and so forth. So now the Allen Institute has released these plans for this thing called it's an optical coherence tomography scope you can build for about $7,000. And so Cameron paid for it, and we got all the parts sitting on a shelf in my lab. We're going to put it together at some point. But it's a thing where you can clarify the brain, and then put it in this little chamber, and there's a light source on one side, and then a camera, and then it rotates it, and it's like a CT scan, where if you image a transparent brain from every angle, then you can computationally reconstruct this full 3D volume and see where the probe tracks are. And it's sort of like light sheet, except that it's cheap and it's fast, and it doesn't give you cellular resolution, but you don't need cellular resolution to localize a probe track. So. Or you can use light sheet, I guess, be the other way. But that's kind of overkill. So, okay, so we'll talk about neuropixels a bit. So neuropixels they're kind of like the hot new thing now. There's these, you know, silicon probes. So some weird decisions were made in their, how they're made, but I think they're still useful. So they decided to just have one giant shank instead of multiple small shanks. So multiple small shanks are good for certain structures like hippocampus, where you have a thin layer of cells and you put all the shanks in it. This was designed really for multi-region recording. So the idea is that you stab that thing through as many brain regions as you can, you just record all of them. So it's less designed for, so the old-fashioned way of doing neurophysiology is like I work on the thalamus. You just put electrodes in the thalamus and only the thalamus, you don't think about anything else. And so your goal is to get as many electrodes into the thalamus as you can. And the philosophy here is more that you just record from a lot of different brain regions at the same time, and then you try to look at how they coordinate their activity. And I think that's a more sort of modern approach. I guess what I'm saying is the old approach was based to a large extent in technical limitations. So what they do, the NeuroPixels probe, it has 1,000 or 960 sites or something like that, but you can only record from 384 of them. So almost everyone just picks the bottom 384. So you have the centimeter long shank, but then you record from the bottom 3-point-something millimeters. And you can see where they are. So the site impedance, so that's one of the things, the older generations of silicon probes, they're typically 1 megaohm site impedances, whereas here you can see it's much less. So the quality of the signals from these probes is very, very good. They're really not that different really than any other silicon probe. They're just kind of longer and they're a little cheaper and they have good site impedances. And so the density of sites is very high all the way along here. I actually wish I brought an image showing what recordings look like, but so what they're good for is head fixed animals and recording from multiple areas simultaneously. You can try to get, so people have gotten up to 8 probes per animal. So that's, and you can record something like 3,000 neurons simultaneously doing that. Problem is that it takes so long to put 8 probes in the animal that the animal is not going to sit there for 2 hours and then perform a behavior after that. So it's, people typically, I think, do more like 3 probes max for behaving animals. But you can get very good quality recordings from multiple brain regions. And then this also, so the new generation ones have a site down at the bottom that allows you to lesion. They have one big site you can lesion with. But with a probe like this that has sites all along it, you can actually get your anatomical localization, the depth dimension, you can get pretty well, because say that that's cortex, then you have some white matter there, and then you have, say, like striatum or something, you have some other stuff. So you can actually see that there's spikes all along here, and then you have the zone where you have no spikes, you have more spikes there. And so you can actually use that to get your depth information as to where your probe is. So in a way, that's the most accurate method of all, probably. Okay, so how do you check where your electrodes went? What time is it? Okay, we're getting close to time for break. I don't know much to say about LP and EEG recordings. It's just you stick a big electrode somewhere and you record an average potential. You know, so things like an EKG, right? You just put these electrodes the size of, you know, bigger than quarters on a person's chest, and you can record field potentials from the chest wall. Or EEG, you know, people put 21 electrodes, whatever it is, on the scalp, sometimes more than that. And EEG are recording from a lot of different sites at the same time, but each one is averaged over a larger area. So what this is good for us, maybe let's talk, well, how about we take a break and we'll come back and talk about this a little bit more. Okay, any questions before we break?
The neuropixels, Luke, is it?
Yep.
So for what type of animals are currently what animals are being probed to use this? Is it just for rats or?
So it was really designed for mice and rats, primarily I think for head fixed mice. People use it in chronically implanted in rats. People have done it in monkeys, but it's kind of too, it's not good for monkeys. So they're coming out, the new version, they have one called primate pixels that has something like 4,000 sites, and it's really big, and that's for monkeys. It has to be stiffer for monkeys because the dirt is thicker and it's just harder to get it in and stuff. I mean, people have used it for a lot of other organisms too.
Okay.
Can't do it in humans though, so that's a problem. But I mean, that's a regulatory thing. So you can't insert something that has to get FDA approval, and then even then you can only insert it into a brain region that you suspect is going to be resected, typically during epilepsy surgeries. So I don't think there's ever been neuropixels recordings in humans, possibly ex vivo brain tissue.
Hmm.
I have a,
Sorry?
keep going.
No, that's all.
This is like probably a really simplistic question, but when you were talking about like how tetrodes, you can move them around if you're in like a bad site. I'm just curious, is a bad site anything more complicated than just like a quiet site or a site that's not?
Yeah,
like,
just a site that's quiet. I mean, so
okay.
One of the things I should say is when people started doing recordings with tetrodes and silicon probes, I think that was to some extent when people realized that the old approach with single wires where you fish for a neuron was really oversampling the high firing rate cells. And and then when people started doing this type of recording where they stick their things in and then you just record for a long time and then you go back and you automatically sort and see what you have, and people found that there's all these low firing rate neurons that you don't really notice. So there are times I do, like if you record in say hippocampus, you see lots of spikes, and so you get your cells afterwards and you're like, okay, I had a lot of spikes. But then there are a lot of cells, but then there are a lot of times that there are these cells that didn't fire that many spikes, but there's more cells than you thought in there, and just a lot of them were ones that didn't fire that often. Recordings in striatum, for example, where it's a lot quieter, I've done say 64 site recordings in striatum where I look and I'm like, I've got nothing. This recording sucks, and there's like one little blip and then there's nothing there, and it's recorded for like 2 hours, and then I spike sort and I have like 50 units, like 50 well isolated units. And it's just because they spike at 0.1 hertz. And in 2 hours, you can get enough spikes to sort them. So, and then when people started doing calcium imaging, they realized that it's even worse than people thought, and that there's a whole bunch of cells that just don't fire at all. And so all extracellular recording oversamples high firing rate cells. It's just that these sort of newer, triangulation-based methods where you have multiple sites per cell, those do it less. But still, it's the case that we're oversampling high firing rate cells. That's something just always keep in mind. Okay. All right, let's take a break. Start recording. Okay. So I don't have any more slides. I just have these. So wait, that's not what I wanted. Okay. All right, so LFP and EEG recordings. So wait a second. Sorry, give me a second. I can only see Claire. All right, can everyone see okay? Works? I can't see anyone, so I don't know what you're saying. Okay.
It's good.
Okay, great. All right, so so the idea here is to record local field potentials, and what that is that if you actually look at real data, I thought I had a slide on that, but I guess I don't. Real data, it looks, you have this sort of things like this, and then you have actual potentials on top of that, right? And so recording the action potentials, we talked about this before, you typically high pass filter, and then you get this thing that looks just like that, right? Or you can low pass filter and basically get rid of the spikes. But actually those fluctuations up and down, that is what's called local field potential, and that carries information. And what it's typically believed to be is information about how, that largely reflects how much synaptic input that structure is getting. So an example would be I don't know, let's go back to hippocampus because it's sort of easiest, but in hippocampus, you have the theta oscillation at 8 hertz. The idea there is that cells are being subthreshold depolarized at that frequency. Or you have the sharp wave and a sharp wave ripple where you have the pyramidal cells in CA1 are upside down, so they're pointing down because the brain, so hippocampus is part of cortex, that's sort of where my fingernails are, and then it goes like this and it curls under so that the pyramidal cells are kind of upside down relative to the top. And so when you put your electrodes in and you're in the pyramidal layer there, you see a ripple oscillation there, but then you go down there, you see this sharp wave, which is the depolarization of the dendrites by the inputs from CA3. So and there's lots of analysis people can do with these things that's super sophisticated and can pull out all sorts of interesting stuff. Like I said, mostly about the inputs. But anyway, so you want to be able to record those. And so they are in different frequency bands. And typically it's the case in the brain. You have things like delta, which is sort of like 4 hertz, theta around 8, beta, alpha, and gamma is a little bit higher, and high gamma, all that. And some people have what they call epsilon, which is the highest, which is really just spikes, or it can be multi-unit activity. So multi-unit activity meaning essentially unsorted activity. You just have an electrode where you're recording many action potentials on it. You don't know which cells any of them are coming from. That's multi-unit, as opposed to single unit where it's isolated and you can identify each cell, right? So the idea here, though, is that you have these different oscillations, and the spatial extent, like the spatial extent to which that oscillation is synchronized within the brain, typically is the case that low frequency oscillations are synchronized over larger parts of the brain, and then high frequency oscillations are smaller in scale. So low frequency oscillations are often, over large chunks of brain. So the challenge, well, so what I'll say is that, so LFPs, if you stick an electrode in there, you'll see LFPs. And if you have multiple electrodes, you can sort of see that sometimes, so here's an example where this oscillation and the ripple oscillation and pyramidal layer is very localized to just that layer. But then if you have the theta oscillation, you can see that everywhere. And it's actually a problem that it's like, it expands too far out, right? So EEGs recorded from the scalp, EEGs typically only see low frequency activity if you're not having a seizure. And the reason why, interestingly, it's not because, so some people think that it's because the scalp is filtering out, it's essentially low pass filtering. And I think this is somewhat controversial, but at least my understanding of it is that it's not primarily that the scalp is low pass filtering per se, it's that the scalp is doing spatial averaging. And because the high frequencies in a non-epileptic brain, all the sort of sources of high frequency stuff are all desynchronized and it all cancels out, because you see low frequency oscillations. And then in seizures, you see higher frequency oscillations, which is true empirically, because the idea there is that in a seizure, you have larger chunks of cortex that are synchronously oscillating at high frequencies, and then it comes out through the scalp.
Hey, Luke. Is it also the case that high frequency oscillations don't travel as far as low frequency, or no?
No, so that's what I'm saying, is it doesn't seem that it's because they don't travel as far so much as that it seems that they're just actually not, that they just don't have as large of a spatial extent, which is, interesting. So, okay, so then we're going to talk about referencing, because this is actually relevant. So the very first, I think it was Joe asked a question about grounding and so forth. So we're going to talk about that now, which is, all right, so referencing. All right, so remember I told you in lecture 1 that voltage is a difference between the voltage at two different places. There's no such thing as a voltage. So as just like one point in space. So if you record an action potential, you're recording the difference between the intracellular voltage and some voltage somewhere else. So imagine you just have like a dish, and then you have sort of neuron in there, and you patch onto it. And what typically is the case, you have this bath solution, and then you have one electrode in there, and then you have your thing, and then you have a second electrode that goes in there and then is in the solution. So you're just measuring, your reference is out there. So reference and ground are not the same thing. They can be the same thing, but they're not always the same thing. So actually the reference is the thing that you're measuring the difference between your electrode and a reference. But the reference is not necessarily at ground. Okay, so grounding I'll talk about next. But so you can have a reference in like a bath solution, you can have something like that, right? So that's pretty simple. In the brain, actually in the brain, it can be a little bit more complicated. So the reason why it can be complicated is if you're recording local field potentials, remember I said that local field potentials can actually, is what's called volume conduction. They can actually conduct away from the location, and you can see them over a larger area. So if you record in, say, cortex right above hippocampus, and hippocampus has really strong theta, then you'll see theta in cortex. And you'll be like, oh, there's theta in cortex, because it's not coming from cortex. And actually, that's an important thing to recognize. So a lot of papers where people do LFP recordings where they only record LFPs, they've come to very wrong conclusions, because they're like, oh, well, the theta in cortex synchronizes with the theta in amygdala or something like that. And then both of those are volume conducted from hippocampus. And the best way for you to test that is you have to record units there as well and look to see if the units phase lock to the oscillations. So if you record in some structure, for example, in ventral striatum, it has this crazy gamma, this huge amplitude gamma. And none of the units there phase lock to the gamma. And people have done all these papers on, and then recently someone looked very carefully with silicon probes where they can get the geometry and they know exactly where they are. They found that all that gamma was volume conducted from nearby piriform cortex, so it's factory stuff. And so, but the big clue is that very few of the units there showed any sort of meaningful synchronization to it. So that and if those were the local, the synaptic inputs coming to that local area, you would expect them to. Anyway, so that's volume conduction. So this is one of the problems with referencing, is that where do you put your reference? What part of the brain is electrically silent, right? So I always put my reference electrodes in the cerebellum, because the cerebellum doesn't do anything. It's electrically silent. We all know that. Except, Cameron would, be very angry if he found out probably I was doing that. But the point is that if you, so say that's the animal's brain, right? And then, or the animal's skull, I should say, and then, I don't know, so you got hippocampus there, you got cerebellum back here. So the thing is, cerebellum's in the posterior fossa. It's actually a different skull compartment. So you don't see as much theta there. So if you're recording, for example, hippocampal theta, if you have your electrode there, and say you had like your reference was up there, and it's a screw on the skull surface, well, that's still going to get a lot of volume conducted theta from hippocampus. So it can be all over that area. So you have to know where your references are when you do things like record, or say, for example, people have put their reference electrode up there, and then recorded, with an electrode there, and they're like, oh, I'm seeing all this theta. And it's like you're picking up theta on your reference electrode, volume conducted from hippocampus. So this stuff is very tricky. So I should say that is when you do referencing for local field potentials. For spikes, it's much easier, and the reason why is that so there's two ways you can do referencing for spikes, and one is to do something like this, where say you have a reference electrode there, and you're recording there, and then you'll get some sort of waveform like that, and that has spikes on it, right? And the thing is that the stuff that gets volume conducted and so forth, it doesn't really matter if you're just high pass filtering and looking just at the spike waveforms. It makes very little difference. The one area in which it does make a difference is that EMG noise. So there's all these muscles that attach onto the skull, and those muscles in the animal chews or moves its head, you see a lot of electrical noise, and that electrical noise is very similar across channels. So if you reference between, say, some distant site and a reference electrode, you'll see lots of EMG noise. So what people will often do is that they will make their reference be one of the electrodes is very close to the recording site, but not exactly at the recording site. So people who do tetrode recordings, for example, will put one tetrode in the white matter right above their structure, and then stick the other tetrodes into the structure. And then they look at the voltage difference between the tetrodes in the structure and the one right outside the structure. The idea being that EMG noise will be approximately equal on all those, and then so you essentially canceling out the EMG noise. And that's called local referencing. So the thing is, you don't have to, so recording systems, some of them you can actually set it up so you choose your reference and you can do that. But you can do what's called local re-referencing instead, which is that you just record everything relative to, say, some distant reference, and then you can post hoc in software go back and take your reference channel and just subtract that from the other channels. And so you've essentially re-referenced your channels relative to that local reference channel. So local re-referencing. Did people, that was a little bit fast, I went over that. Did anybody have questions about that?
Do people ever reference from multiple electrodes? Like, for example, you wouldn't, like after recording, you would just take the average of a group of electrodes and then?
Yes, exactly. Yep, that's another way to do it. That's a good question. So typically what you would try to do is, let me put it this way. So if you had a tetrode, you wouldn't re-reference to the average of those 4 channels because there's at least going to be some of that spike content in those 4 channels. So what you might do is you might re-reference to the average of the 4 channels on the adjacent tetrode.
Okay.
Right, but you wouldn't necessarily re-reference to actually ideally you re-reference to sort of the average or median of several other channels. So you could with multi-shank probes, I would typically re-reference to the median of the median of all the other channels on different shanks, for example. So
It would have to be like at the same time, right? Like you wouldn't, for example, you wouldn't say like have a recording from previously when like an animal is not performing a task and then reference to that recording
right.
like later.
Yeah, so that's a good question. Let me explain a little more what it is that you're trying to get rid of. So the idea here is if you're just looking at spikes, so say that you have a recording where you have multiple channels go like this, and then the animal in the middle starts chewing or something. Then it's going to go like that, right?
Mm-hmm.
Then this channel is going to look like this. And they all, but the thing is that all of these look almost exactly the same. And so imagine that so what you can do is that if you have spikes on this channel, right, well, if you just look at that, you can't, or it'd be difficult for you to isolate the spikes out of that mess there. But if you have an adjacent channel, say in the white matter or something, that looks almost the same, it has almost the same EMG artifact, but no spikes, then you essentially take, if that one's A and that one's B, you can do A minus B, right? And that's local re-referencing. So what you're doing is that simultaneously recorded channels so the simultaneously recorded channels will record the same noise as your channel of interest, but it won't record the same signal. So it's a way of sort of recording just the noise and then subtracting it out afterwards.
Okay.
And so like I was saying, some people will, in old systems would do it so that the system is actually configured to do that in hardware, but I think it's better to do it just post hoc.
Luke, can you talk about local versus median re-referencing?
Well, median re-referencing is a type of local re-referencing. Okay. So I guess it depends on the number of channels you have. So the idea is if you have a tetrode, you wouldn't want to re-reference to the median of that same tetrode because spikes are going to be visible on all 4 channels of that tetrode, or on 3 out of the 4, something like that, right? So what you want to do is you want to re-reference to adjacent channels where the spikes are not visible. So if you have a 64-site probe, then where say 4 sites pick up a spike, then you could re-reference those 4 sites to the median of the entire, or you can re-reference each individual site on that probe to the median of the probe, but that's because there's no spikes that will be picked up on all 64 sites, or even more than a tiny fraction of those sites. So it's just a question of how many sites you have, and how many sites you have and how many sites you expect spikes to be visible on. Make sense? Okay. So, but the reason I would say local re-referencing is a better idea than just recording with the local reference is because if you just record with the local reference, then you're not getting LFP information. So, so the idea here is that you record LFPs and spikes on the same electrode, and then you get your LFP information, and then you can locally re-reference to get the spikes. So, So that also has to do with data acquisition and the sort of technical aspects of that. And I'll explain why. So in the old days, the old days, 90s and before, the way that people did this, at least my understanding is that so what you would have to do is if you wanted to record action potentials, you would do something like local re-referencing, and you have to do high pass filtering all in hardware, right? And then you get some sort of signal, you know, that looks like, not drawing very well, but has action potentials on it, right? And what you would do is you pass that through an oscilloscope and you watch it. And what you'd see on the oscilloscope screen is so what oscilloscopes do what's called, so an oscilloscope plots voltage as a function at time. And it typically works in trigger mode, which means that every time it detects some voltage crossing some threshold, it takes a snapshot just of that and it shows it to you. And so what you do is you'll look at the screen, and then you'll have some sort of trigger that you can set, and you set it to the point where you get the thing like that, where it's capturing just those spikes, not other stuff. And then you essentially set a hardware trigger where you're like, okay, and then you just basically record a timestamp every time the voltage crosses that line. And then you just record like a blip, right? And then so you're not actually even recording the waveforms and stuff. Or the way that those systems really worked, I think maybe that was the first ones. Then later what they would do is they would have some sort of system that would sample just the image of the spike at each time the thing was triggered. So this data is not recorded. You're only recording the action potential waveforms themselves. And this was technical limitations. I don't know, so they had a system like this in the first lab I worked in college. It's a very old setup, and they told me they were all excited about the time that they bought a 256 kilobyte hard drive, and it was the size of a dorm fridge. And so like back in those days, yeah, you couldn't do 128 channel recordings at 30 kilohertz and just save the whole thing. So they had to use these tricks to be able to shrink the data down to something that's manageable. So something like this, you kind of have to use local referencing, because you essentially have to you have to isolate the spikes in hardware prior to sampling them. You can't just record the whole thing and post hoc in software fix it. So a lot of commercial recording systems still use this, where they'll actually record, where they'll record an LFP signal at a lower, so the LFP, the frequencies in the LFP that you're interested in are all, say, below 300 hertz or something, 200 hertz, whatever. So it doesn't make sense to sample at, say, 40 kilohertz. Whereas with spikes, if you want to have a good picture of the waveform, you sample at least 20 kilohertz. Remember we talked about it's about 8 kilohertz is the top of the frequencies that are present in a spike. So if you sample the entire thing at 20 kilohertz or more to get your LFPs, but then you're vastly oversampling the LFPs, which only have frequencies below a couple hundred hertz. So the way that a lot of these commercial systems will still work is that it'll sample the LFP at a low sampling rate, and then it will sample the spikes at a higher sampling rate, but it'll only save the spike waveforms themselves. So that's one way that people can do it, and that saves disk space and stuff. So the point is, but then you have like LFP at, say, I don't know, say 1 kilohertz. Then you have spikes at something like 30 kilohertz. Okay, so that was done in the old days for disk space, and then I think some of the newer systems, the way that they work is they just sample everything at 30 kilohertz or 20 or 40 or whatever it is, and then you just reconstruct everything and you save everything. You just buy a lot of hard drives. And what you can do there is you can, if you want, so what we typically do, for example, is extract do all the processing on all the spikes and sorting of the waveforms and all that, and figure out what the timestamps are of the spikes, which ones they correspond to, which units they are, and then just save that in like a MAT file from MATLAB. And then you can take a 200 gigabyte data set, and then just the spike, the spike times and identities will be like 20 megabytes, right? And then you can take the LFPs and downsample them down to 1 kilohertz, 1.25 is what we usually use. And then your data is shrunken down by a factor of 20 or so. So, and sometimes we don't even necessarily keep LFP on all the channels. I mean, so we don't throw the data away, but what I mean is we sort of take it and then you purify out the stuff that's relevant from it. And then you just put that old data on a drive or tape somewhere, and you put it on a shelf somewhere in case you ever need to access it again. But then once you've extracted the stuff you need, then it's actually much, much smaller. And that's the way most stuff, I think, in biology works, is that whether it's cryo-EM or, 2 photon microscopy or whatever, that people generate these giant data sets, or you know, RNA-seq or whatever. And then you generate a big sequencing data set, and then you figure out how much of each RNA is present, and then that information is actually tiny. All right, so that's one way to do it. You just sample everything, and then you extract out spikes and LP. So the neural pixel system frustrated a lot of people on the way that they chose to do this. And I'll explain why and how they did this, because it was based on sort of hardware considerations that we talked about earlier. So the signal coming out, so the problem with the NeuroPixel system is that because they have so many sites, so if you're recording 384 sites simultaneously, you have to somehow pass all that information into the computer. And there was some, hardware-wise, there was some limitation on the number of bits per second they could send. And as a result of that, it had to be, and I think that's what it was that they do 10-bit sampling. Okay, so what 10-bit sampling means is sorry, I'm gonna pick on someone. So how many levels, if you have 10-bit sampling, how many different voltage levels can you represent? I'll just pick on someone if nobody's gonna volunteer. Is there only 6 people here? Oh no, okay. All right, Jaslyn, what do you think?
Sorry, we don't know.
So if you do 10-bit sampling, okay, so all right. Rowan, you know the answer. What is it?
2 to the 10th.
Okay, good. Who said that? I didn't see who.
Victoria.
Okay, good, Victoria, perfect. You have 2 to the 10th, right? So what is 2 to the 10th?
I need my calculator.
It's 1024. Now, why did I write 10 to the 24? This 1000, damn it. That's not enough. I'm actually gonna erase it. It's 1024. All right. So if you imagine that, if you have 1024 levels so they can only represent 1024 different voltage levels, right? So if you actually look at real data, remember I told you it looks like this. Right, so if you represent the entire, you know, LFP fluctuations, and so say if your range 1020, you know, so if you're 1024 levels spanning that, then the number of levels that will actually be available to record a single action potential, you know, your action potential is gonna look like this, right? So it's gonna be useless. So what they ended up having to do was they had to split it into LFP and spike channels, where the LFP channel is low pass filtered at some frequency and then sampled at probably a low rate, like 1 kilohertz or something, and then the high pass filtered channel, they don't record just the spikes, but so the high pass filtered version of that, you know, I shouldn't say it's not noiseless, but then, you know, then you have spikes on it, right? And so that, if you stretch your 124 sites or you know, voltage levels over that, you can get adequate sampling. So so then they did that, and then this is sampled at 30 kilohertz. So a lot of people were annoyed by that, but I think it's just one of those things. There's trade-offs. You have to decide, you know, what's important and what's not. So one interesting thing about the NeuroPixels format is that, so the reason I told you guys about int16s and int8s and all that, so a common phenomenon that happens is that, all right, so most programming languages have an 8-bit data type, and they have a 16-bit data type, and a 32-bit data type, and that sort of thing. They don't have a 10-bit data type or a 12-bit data type. Those typically just don't exist, so that you can't make a 10-bit integer. So what that means in practice is that if you're doing, say, 10-bit sampling, that you're saving it as a 16-bit integer, and just 6 of those bits are empty, they're zeros. So that these data files are gigantic, and like 6 bits out of every 10 are just zeros. So that's the kind of thing where it's just the way computers are and the way the languages are made, and so that's common. So you can do compression and so forth, and that's the kind of thing that's very easy to compress. Anyway, so that is, I don't know, maybe what you need to know about the data acquisition aspect of it. All right, so we're doing well time-wise. Any questions before we move on from this?
Yeah, Luke.
Yeah.
So for this data acquisition, when you're separating the LFP and the spikes, you're still recording using the same, like, one recording probe or whatever, but is all the separation is done post data collection?
Yeah, well, yeah, so you have many sites, and then each, so you know, one site is going to, you know, look like that, right? And then the next site will be, you know, maybe looks like this. Say that you have sites look like that, and then there might be a spike that looks like this. Right, so you just record all the sites, and then you go back and use your sorting software and says, well, this one is different than that one. And then you
Right.
put neurons from there.
Okay, I was just confused about the LFP versus the spikes.
Oh yes, the LFP is saved in a different file, and you don't use the LFP information when you're spike sorting.
Right, but so you collect LFP and spikes from the same probe though. Like
yes, exactly. Yeah. Yes, exactly. So the data from the analog signal from the probe is actually split into two separate channels. One is high pass filtered, one is low pass filtered, and then the high pass filtered one is sampled at a higher frequency than the low pass filtered one is.
Oh.
But the idea here is that the reason for doing it wasn't to save disk space. Because actually they're sampling the full 30 kilohertz. So it actually takes more disk space than if they had just sampled the entire thing at 30 kilohertz.
Right, right.
But the reason for doing it is because of the 10-bit A to D conversion. And because, so essentially the dynamic range is not good. Whereas, so if you use like one that has 16-bit A to D conversion, you just record the whole thing at 30 kilohertz and you just save the whole thing. So this is just another hardware limitation.
Got it, thanks.
So anyway, this is the kind of thing where it's important to know this stuff, because if you work with data like this, well, I mean, not that all of you will, but that's how it works. So, all right, so grounding is the next thing. So we talked about referencing, now we're gonna talk about grounding. So the idea is that there's 60 hertz noise everywhere, right? And there's all the lights and all stuff running through the walls, and 60 hertz noise, or 50 hertz if you're in Europe or outside the US, then that's this sort of bane of every electrophysiologist's existence, right? And you want to try to do things to minimize it. So I've seen labs that that run entirely on car batteries. So they're like these labs that do single channel recordings, and they just have like a room full of car batteries and chargers, and then they're charging all these car batteries, and they carry them in, and all the rooms, the lights are all turned off, and all the computers are plugged into car batteries, all their, you know, everything's battery operated. And batteries are actually the lowest noise power source that you can get, pretty much. They're almost noiseless. So that's the extent to which some people go. Other ways you can do shielding. So some people will actually have a whole room that's a Faraday cage. So essentially, if you put something inside a Faraday cage, so Faraday cage would be, you know, so say that you have all the 60 hertz out in the world, and if you build some sort of conducting cage, and then this thing goes to ground, right? Then whatever's inside here doesn't have 60 hertz noise. So ways you can do that, you can, some people will actually build a room that costs a ton of money that has metal in the walls and the whole thing's grounded, and then nothing goes in that room that generates 60 hertz noise. Another way you can do it, you can get this conductive silver paint, and you can paint the walls of your room and your ceiling and everything with this paint. It's expensive paint, but it's still cheaper than building a whole room and then ground the paint. So there's other ways you can do it where you can just say put the experiment in a box that has, you know, a ground shield around it. Another way you can do it is you can actually, if you have the animal's head, so for freely moving recordings, people often do like, this is what my lab typically does, animals like that, and then you build a shield like that on top of the animal's head, and so all your electrode stuff's in here, and then this shield is made out of, conductive copper mesh, or sometimes people make a plastic one and then silver paint it or put aluminum on the outside, aluminum foil. And then that thing's grounded, right? So that's ways you have to do shielding first. So to shield things, then the shield itself is grounded, it's a ground, okay? So then shielding's one thing. Another thing is with grounding is ground loops. Okay, so ground loops are a pain. And so what I'll say about shielding and grounding your setup and everything is that it's 50% based on the actual principles of how all this stuff works, and it's 50% just total voodoo and empiricism. You just have to do stuff until it works. So what ground loops are is, so if you remember from physics that if you have a loop of wire, you have in a magnetic field that's changing, then it induces a current through the loop. And so that's how things like generators work. So the idea is that if you, so the notion of a ground loop would be that, I'm trying to think of a good example so I can draw one. So say that you have your, computer there, like that, and that computer is grounded. Then that has a USB port that that goes to some sort of piece of equipment and plugs it in there. Then this thing has like a TTL input that comes from your, I don't know, so say you have like a amplifier. And then that sends some sort of TTL pulse there. And then this thing plugs into the wall and is grounded. So, all right, so now you can see that you've got a loop, right? You have something where this thing is grounded in multiple places. And so, what happens when you say make that connection there is that now all of a sudden you've got 60 hertz noise all over this place, over the place. And so that's a ground loop. Okay, so the way to try to get around ground loops, so there's ways you can do. One thing you can do would be to, so you want everything grounded through one point, at least everything that's electrically connected to other stuff, you want it all grounded through one single point. So one way of doing this would be you could use like a USB, so there's what's called opto-isolators. So an opto-isolator is some sort of thing where you put an electrical signal in and it just converts it to, say, light pulses, and then there's essentially a photodiode on the other side, and then it converts it back into an electrical signal. So you can get, for example, USB opto-isolators, or you can get ones for things like TTL signals where you pass through and then it passes the signal through, but it breaks the electrical contact. So it's easy, it's very difficult to do that for an analog signal, but easy to do it for a digital signal. So that's one way to do it. Another way to do it is to essentially unplug one of your things from ground, right? So if you have a computer or say your amplifier or something, so something is typically grounded if it has a electrical plug that has 3 pins on it. So the third pin is the ground pin. So typically then that thing is grounded. So what you can do is you can buy those things. You ever seen those adapters for old outlets that only have 2 prongs, and you plug it in and it has a 3rd prong, and then you can plug a 3-prong, plug into a 2-prong thing? So if you do that, then your thing is no longer grounded. So if you do that with your CPU, and you essentially then your CPU is floating. If it's not grounded, that means it's floating, meaning its voltage can fluctuate up and down, and you're essentially grounding it through the USB port, the TTL, and the amplifier down into there, right? So that could be dangerous if you have certain pieces of electrical equipment that, for example, say that then you unplug the USB port or USB cable or something, and then your computer might go to some voltage, just drift up to some voltage, and when you touch it, you get shocked or something. So you have to be careful with doing this, but that's another way you can do it, is to essentially, as long as stuff is grounded somehow, it'll work. And then what is typically recommended for this type of thing is that instead of, people will often ground this to that and ground that to that and ground that to that. The better way to do it is if you have some sort of central ground that comes there and then ground each thing separately in a star configuration instead of a loop configuration. And then that way everything is grounded to one point. And that's the best way to do it if you can. Like I was saying, you just largely have to experiment. So whether you short the ground to the reference or not, I actually usually just connect the ground to the reference and it's usually fine. But okay, so here's an example. If I had an animal on a metal table, so that the animal was making electrical contact with the table, I would never do that. But then you'd have to ground the table to get rid of 60 hertz noise. But if I did that, I would never ground both the animal and the table, because then you're gonna have a ground loop, right? So the way you could do that is you could say put the plastic piece of the animal doesn't touch the table, right? Or at the very least, you'd want to record with a separate ground and reference. So what I would typically do in a case like that, so normally the amplifier I use actually just connects the ground and reference together, and then you can separately ground the table just into the wall, but then make sure that the animal doesn't touch the metal table, for example. So the point is that with lots of setups, you have to do grounding, or I mean, you have to figure out ways to do grounding that you don't get 60 hertz noise. So, and you just have to understand the principle of not having loops, opto-isolate if necessary, whatever float things if necessary, and then you just experiment until you get it to work. So this is much more of an issue for intracellular recording than for extracellular recording, I should say. But then there's also sources of noise, like if you're trying to record on a 2-photon microscope, or something, then all the like scan head and all that stuff is making all sorts of crazy noise, and you have to figure out ways to shield from that. Questions about that? I think that's probably the end. So keep the ink and I'll post it, but anybody have questions about any of that stuff? Or electrophysiology in general, or recording or anything? You look dismayed, Claire. No, not dismayed.
Just my face.
All right, so I know it's a lot of information. So next week, I'm going to be not here, but it'll be my student Ellie and my postdoc Suyan will be leading a tutorial for all of you on extracellular recording and spike sorting. So the process of spike sorting first is that you somehow get your raw data and you extract the spikes, the things that are putative action potential waveforms, and then apply different algorithms to sort them so that you can try to cluster them into clusters that likely correspond to different cells, and then going through and then analyzing those and seeing which ones you think are actual cells versus groups of cells versus just electrical noise or muscular noise or some sort of artifact, and then figuring out how you can split and merge clusters until you get something that you think is a pretty good representation of where you have clusters that correspond to things that are probably real neurons in the brain.
By tutorial, do you mean we will need any software or anything, or we'll just watch them?
I think just watch. I don't know. I don't know if a good way to. I think it'd just be easier if you just watch, but you'll have to participate more than you.
I just want to know if I have to download anything.
I thought about that. I don't think so. I think it's just easiest if you just watch. I mean,
okay.
That's the sort of thing where if we did it hands-on in real life, then we could probably do that. But what's going to happen is like installing the software, there's going to be some sort of problem, and I don't want to make it a requirement to do that. But it will actually require actual participation. So you'll have to call out what do you think is a spike or not, what do you think is noise or not, get some ideas about that. Okay, all right, well, if that's all, then I guess I'll see you on, so next week, and on Thursday, I'll be gone. It'll be about that. And then on Tuesday, it'll be about biophysics of opsins and indicators. Okay?
Great.
All right. Bye, everyone.
Bye.